Hello everyone, welcome to this video on antibiotic resistance. My name is Angama Joshua and today we'll explore what antibiotic resistance is, how it develops and the different mechanisms that bacteria use to evade antibiotics. Understanding these mechanisms is crucial as antibiotic resistance has become a major global health threat. So let's dive in. We are going to follow the outline on the right of your screen, so you can always look it up to find where we are at. Antibiotic resistance occurs when bacteria develop the ability to survive exposure to antibiotics that would normally kill them or stop their growth. Essentially, the bacteria outsmart the drug. This is especially dangerous because resistant bacteria can continue to multiply and spread, leading to infections that are much harder to treat. For example, methicillin-resistant Staphylococcus aureus, or MRSA, is resistant to many antibiotics that we commonly use. This means more complicated treatments, longer hospital stays, and in some cases, higher mortality rates. Just to lay the groundwork on why this topic is important, this is an antibiogram of a pus culture from a patient's wound. Here, the main germ responsible for the infection was Escherichia coli. The strain of the bacteria was resistant to almost all of the antibiotics we tested, as we can see denoted by the R's, except for two, imipenem and cotrimoxazole. Antibiotic resistance develops primarily due to two mechanisms: natural selection and horizontal gene transfer. In natural selection, random genetic mutations that make bacteria less sensitive to antibiotics can give them a survival advantage when exposed to the drug. Over time, these resistant bacteria become more common. Horizontal gene transfer, on the other hand, allows bacteria to share resistance genes. This can happen in three ways: transformation, where bacteria pick up free-floating DNA; transduction, where bacterial viruses known as bacteriophages transfer resistance genes; and conjugation, where bacteria exchange plasmids, which often carry multiple resistance genes. There are four mechanisms through which bacteria resist antibiotics. We have enzymatic degradation of the antibiotics, alteration of target sites, efflux pumps, and reduced permeability. Let's look at each of these in detail. First, bacteria can produce enzymes that break down or modify the antibiotic, making it ineffective. One common example is beta-lactamase enzymes, which destroy the beta-lactam ring in antibiotics like penicillins and cephalosporins. This is particularly concerning because beta-lactamase producing bacteria are resistant to a wide range of antibiotics, forcing us to use more potent or toxic alternatives. Next, bacteria can alter their target site, the part of the bacterial cell that antibiotics attack. A good example is methicillin-resistant Staphylococcus aureus, or MRSA. This bacterium modifies penicillin-binding proteins, or PBPs, making beta-lactam antibiotics like methicillin and oxacillin ineffective. Other bacteria can alter ribosomes to resist antibiotics like macrolides or aminoglycosides, or change DNA gyrase to resist fluoroquinolones. Another mechanism is the use of efflux pumps. These are proteins in the bacterial cell membrane that pump antibiotics out of the cell before they can reach their target. Efflux pumps can often expel multiple types of antibiotics, making bacteria resistant to several drugs at once. For example, the ACR-AB Tol-C efflux pump found in gram-negative bacteria can remove drugs like tetracyclines and fluoroquinolones. Bacteria can also become resistant by reducing the permeability of their cell membrane, which limits the amount of antibiotic that can enter the cell. Certain bacteria like Pseudomonas aeruginosa modify their outer porins to prevent beta-lactams or carbapenems from entering the cell. When bacteria reduce antibiotic entry, Higher doses are often needed, which can be more toxic to the patient. In some cases, bacteria combine multiple resistant mechanisms, making them extremely difficult to treat. A prime example is Acinetobacter baumannii, which produces enzymes, alters target sites, reduces permeability, and uses efflux pumps. This combination makes it resistant to nearly all antibiotics, earning its label of a superbug. So how do bacteria acquire these resistant traits? The answer lies in their genetics. Chromosomal mutations occur naturally during bacterial replication. Plasmids, which often carry resistance genes, can be transferred between bacteria via conjugation. Transposons and integrons, mobile genetic elements, can carry resistant genes. And bacteriophages can introduce resistant genes through transduction. This genetic flexibility allows bacteria to rapidly adapt and spread resistance. It's also important to distinguish between cross-resistance and co-resistance. Cross-resistance means that when bacteria becomes resistant to one antibiotic, they can become resistant to other antibiotics in the same class. For example, resistance to one beta-lactam often means resistance to all beta-lactams. Co-resistance happens when bacteria acquire resistance to unrelated antibiotics, often due to genes carried on the same plasmid. How can we address these mechanisms of resistance? First, we can use combination therapy, where two or more antibiotics with different mechanisms of action are used together to prevent resistance from developing. Beta-lactamase inhibitors like clavulanic acid can block enzymes that break down beta-lactams. There is also ongoing research into drugs that inhibit efflux pumps. And most importantly, we need to reduce the unnecessary use of antibiotics. Rational prescribing can help slow down the development of resistance. In summary, bacteria have evolved various mechanisms to resist antibiotics, including enzyme production, target modification, efflux pumps, and reduced permeability. These mechanisms can occur individually or in combination, complicating treatment options and leading to multi-drug resistance. Understanding these mechanisms is crucial for healthcare providers to develop effective treatment strategies and reduce the spread of antibiotic-resistant infections. Antibiotic resistance isn't just a scientific issue, it's a global health crisis. Resistant infections lead to increased morbidity and mortality, prolonged hospital stays, and higher healthcare costs. For example, carbapenem-resistant Enterobacteriaceae and multi-drug-resistant tuberculosis are especially dangerous. To combat antibiotic resistance, we must prescribe antibiotics only when necessary, implement strict infection control measures in hospitals, track resistance patterns globally, and importantly, invest in the development of new antibiotics to stay ahead of evolving bacteria. In conclusion, antibiotic resistance is a growing global health threat. It's driven by the misuse and overuse of antibiotics, and if we don't take action, we risk losing the ability to treat common infections. Healthcare professionals must use antibiotics responsibly, and patients should always complete their prescribed courses. Together, we can preserve the effectiveness of these life-saving drugs for future generations. Before we wrap up, let's go over a few practice questions to test your understanding of the material. You can pause after every question to think about it. What are 4 mechanisms by which bacteria develop antibiotic resistance? Take a moment to think about the different ways bacteria can evade antibiotics. Explain how efflux pumps contribute to antibiotic resistance and provide an example. Think back to how bacteria use these pumps to remove antibiotics from their cells. Describe the difference between cross-resistance and co-resistance with examples. Consider how resistance to one antibiotic can affect resistance to others in the same or different classes. Hopefully, these questions should help reinforce the key points we've covered today. Be sure to review the material and try answering these questions to solidify your understanding of antibiotic resistance. Thank you for watching, and I hope this video has given you a clear understanding of antibiotic resistance and the importance of combating it.